All right. Welcome, everybody, on this soggy Wednesday that's apparently going to turn into ice. Hopefully, it won't. We'll get you out of here before that. So it is my pleasure to introduce Dr. Chris Kuzawa. He has an extensive CV of lots of accomplishments and awards, so I'll kind of hit the main highlights. Chris got his bachelor's degree in anthropology at the University of Colorado Boulder, and then got a master's in public health and a PhD in anthropology at Emory University. He then received an NIH National Research Service Award to do a postdoc at the University of Minnesota in epidemiology. And now he is the John D. MacArthur Professor of Anthropology, as well as a faculty fellow here at IPR and a founding member of the C2S group here on campus. Chris is also a member of the National Academy of Sciences and the American Academy of Arts and Sciences. And he's done a huge range of work that covers lots of different topics, some of which include developmental origins of adult health and disease, which he has worked on primarily in the context of a study based in Cebu, Philippines, a cohort study that started in the 1980s. He also works on male reproductive ecology and the psychobiology of fatherhood, the evolution and developmental energetics of the brain, as well as issues of intergenerational health and understanding the mechanisms through which health is transferred across generations, among many other things. My understanding is that today he's going to talk to us a little bit about the brain.
Nope.
Not the brain? Oh, never mind. That was another idea that was thrown
time.
about. Yeah.
Don't worry about it.
Anyhow, with that, I will let Chris give his talk. Thank you
Okay.
for being here, Chris.
Right. Thanks, Katie. Thank you for showing up. This weather is just the pits. It really is. And I appreciate you being here. And if you opted to go virtual, I made a good call. Thank you all for tuning in one way or the other. And okay. See if this will work. It apparently is not. Okay. Yeah. So we might need a little bit of tech help here. Nothing is moving the slides. Yeah. Okay, you know what? Maybe the oh, it's working now. Okay. Yeah, good. All right. So when you look across the globe, the biggest causes of mortality are cardiovascular diseases, things like heart attack, stroke, diabetes, et cetera, right? And so here you see that those are much higher than the other lines and that they're increasing as well across time. And this is happening in parallel with increases in body weight. And this shouldn't be too surprising because when you think of these kinds of cardiometabolic diseases, the main factor that you think of as a risk factor is eating too much, gaining too much weight, not getting enough exercise, and so on, right? So these are kind of diseases of excess or of overnutrition. And that's such a well-established idea that when these findings were first published back in the late 80s and the early 90s, they were met with a lot of surprise because what this shows is the same thing. It's the rate of heart disease, but not based upon one's adult weight, but based upon one's weight at birth. And you can see the relationship is the opposite of what you'd expect. It's the lower birth weight babies, okay, who are presumably undernourished that go on to have elevated risk for cardiovascular disease. And this was first really shown by David Barker, epidemiologist from the UK, who passed away a couple years ago, about 10 years ago now. But it's been replicated many times. There's been a ton of work on this. And here's a meta-analysis that pulls data from about 8 million people. And you see that, and this happens to be diabetes, as birth weight goes down, risk goes up, very much like what Barker showed. You also see that over here, there's this increase among the highest birth weight babies. This is something that you see in relation to obesity. So when somebody's obese, there are effects of that on the next generation that elevate risk for disease. I'm not going to talk about that. I'm going to focus on the left-hand part of this slide. And the model that Barker proposed to explain this is this. So first of all, we know that a fetus that's stressed or undernourished is going to slow its growth rate and you get a low birth weight as a result of that, right? But what Barker pointed out, or what he proposed, is that the fetus is actually doing a lot more than that. It's adjusting all these other biological systems, like how the body handles lipids, glucose, where they put fat down, how you regulate blood pressure. And these can be relatively permanent and persist into later life to influence risk for cardiovascular disease. Now, everything I've shown you so far is just birth weight. And you may be wondering, is this really telling us something about prenatal nutrition, right? Because Birth weight is also a function of genes, whether or not the mother smoked. If you're a low birth weight baby, you're born into a lower income household. There are many factors that actually could be picking up that aren't just nutrition. So experimental work in animal models has been key to kind of nailing this story. And what this shows here, these are guinea pigs and their mothers, these right here, the open circles, their mothers had their diet restricted by 30% during pregnancy, right? And you can see that, okay, first of all, this is glucose in the fasting state here. They have a glucose infusion, it pops up their glucose levels. And then their bodies produce insulin and they bring the glucose back down. And those, those animals that were prenatally undernourished are actually clearing their glucose just fine. But when you look at their insulin, you see a different story. So on the fasting state, their insulin levels are higher, okay? And then when they experience that glucose infusion, they're overproducing insulin. So their bodies are clearing glucose, but they're having to work much harder at it, which shows us that their tissues are resistant to the effects of insulin. And this is one of the factors that eventually leads to diabetes. So these kinds of experimental studies in animal models give us a lot of confidence that that this finding of elevated diabetes risk in low birth weight babies and humans is actually something about prenatal nutrition in part. I'm going to show you one other thing, just a quick snapshot of some of our work in the Philippines. I'm really not going to talk so much about our work. This is more of a synthesis of the literature as a whole, but I want to illustrate one other finding that's common in this literature. What this shows again is insulin, okay, so fasting insulin in relation to quartiles of adult waist circumference. So this is telling us how much body fat there is. And as you would expect, fasting insulin goes up. The diabetes risk goes up among the heaviest adults. Now, when you stratify further on the basis of birth weight tertiles, you can see that birth weight doesn't matter too much down here, starts to matter more here, and matters quite a bit here. And the story that comes out of this is that being born small, but then going on to become a relatively heavier adult is the highest risk scenario, right? And the gradient here is pretty big. Like there's about a 25% difference in risk across birth weight tertiles. So what this is an example of is what's called developmental plasticity. And this is the capacity of a single genome to create a range of different phenotypes depending upon the environments that are experienced, right? So you have a lot of possible outcomes. And in my field, and it's really been about 100 years or a little more that anthropologists have been studying this. And one of the things that we're interested in is how this kind of process of plasticity allows adjustment to different kinds of environments. So I'll give you one quick example of that. Like if we were to go to high altitude setting in like Tibet or Nepal or Peru, we're adjusted to low altitude. And so we're going to have this experience, this increase in heart rate. We're going to probably, some of us will get altitude sickness. Our bodies will have to ramp up production of red blood cells and so on. But if you're raised at high altitude, you can actually grow bigger lungs as a solution to this. So if this what this shows is the age at migration to high altitude in Peru. And if you migrated to high altitude at age 2, you have bigger lungs, and that capacity is reduced the older the migration occurred, right? So what this shows is that there's a capacity, in this case, to grow bigger lungs in response to low oxygen pressure, but it declines with age as development unfolds, right? So this is an example of how plasticity allows adjustment to the environment. And so when these Barker findings, showing low birth weight, increasing risk for these diseases of overnutrition, a lot of us were curious as to whether there might be something similar, like an adaptive process that's going on here. Could there be kind of a cue that the mother is providing about the nutritional experiences that the fetus is going to experience, allowing it to adjust aspects of its metabolism, its growth, and so on in anticipation of that? And Pat Bateson in 2001 talked about the mother sending a weather forecast as a sort of example of this. And then Gluckman and Hansen have published a lot on this. Now, the challenge to this idea is the issue of signal fidelity, right? Because we live 70, 80 years, or usually. Does it really make sense to kind of set your nutritional expectations and your metabolism for life based upon the conditions that you're going to experience during just a couple months during pregnancy? What if your mother's pregnant during a really good season or a really bad season during a famine? Do you adjust your whole strategy based upon that? And you can see the problem with that sort of strategy. This is showing 400 years of data on the price of rye in Germany. You can see it's going all over the place. And there are these big spikes here that are related to volcanic events. So when volcanoes happen, productivity goes way down because sunlight goes down, right? So this just gives you a feel for how variable something, these natural systems can be. Oh, I should have given you a trigger warning, but this quote, I think, illustrates a common problem, right, for folks who are trying to convince the world that climate change is changing and that global warming is happening. And I think there's an analogy here. There's the distinction between weather and climate. And you often hear skeptics about global warming pointing to the fact that, oh, it's so cold right now. Where's the global warming? Well, the answer to that is that the global warming is not in any given data point. It's not here, it's not here, it's not here. It's in the trend through the data, right? And that's what climate is all about. It's about sort of, cutting through that variability and that short-term noise. And I think, oh gosh, I had no idea that was, what do I do? What about that stuff at the top? Can we minimize it? How?
More.
Oh, I had no idea that was up there. Sorry. Anybody knows what to do? Tell me. Okay. How about I just like move it off the screen or something? Well, boom. Oh, that's the other one. Yeah, thank you. I had no idea that was obscuring your view.
Click on this again.
Yeah, okay, good. Yeah, anyway, so I think that there's an analogy here. If you think about a fetus trying to predict its future based upon what's going on in utero, it does not want to adjust its strategy based upon any given data point, whether it's born in this year versus that year, but actually somehow track that underlying trend. And that's the main thing that I'm going to be sort of digging into in my talk. Does fetal nutrition convey information about average local nutrition akin to climate? Yeah, sure. What? I'm sorry.
Yeah. Because of infant mortality, so many children would die at a young age. Could there have been, could the response be not for your entire life, but could the response be targeting a much shorter period?
Let me tell the story, and then you can ask that question. Okay, we can address it at the end, because there's a lot that will sort of get to what you're bringing up here. But before I get into this, what I want to do is quickly, okay, so I showed you that model, right, showing that nutrition is driving all those fetal responses, right, the Barker hypothesis. But the other piece of that is stress physiology. The hormone cortisol is a big piece of it as well. So those are both impacting the fetus. They both come from the mother. And when the mother experiences stress, her cortisol goes up, right? And that can cross the placenta and trigger all kinds of biological changes in the fetus akin to nutrition. So here you see, this is sheep. And the point here is that it's exactly the same as what we saw in the guinea pigs, right? So if they're overexposed to maternal cortisol in utero, they're clearing their glucose, but they're having to overproduce insulin and work harder to do it. So you're seeing the same sort of insulin resistance in relation to cortisol exposure as you are to nutrition. Okay, and so I will come back to this. That's something I want to come back to at the end, but I just want to set that up as the other system that we'll eventually address. So what I want to talk about today to explore all this is a couple of case studies. First, the Dutch famine study, which some of you may have heard of. And then there's a study in the Gambia. We'll dig into those. And I think that what these studies show is that there's a lot of maternal buffering of the fetus, and that actually what she experiences is not getting through nutritionally. And then we'll talk a bit about the evolution of maternal nutritional buffering strategies and how this might have impacts on this idea of signaling that I've set up here. And then we'll think about lessons for this other system, the stress physiology. Like based upon what we see in nutrition, does any of that potentially apply to stress biology? And then we'll have a few quick wrap up and implications at the end. Okay, so to begin, just how strong is the evidence that maternal nutrition during pregnancy influences offspring health outcomes in humans? Right, we have all this evidence that under smaller birth weight, babies go under elevated risk. We have a lot of evidence that if you restrict the nutritional intake of rats or sheep or mice, their babies go on to have elevated risk. But what do we see in humans? And the evidence, to steal my own thunder, is not particularly strong. So most of the work in this field that's called DOHED, Developmental Origins of Health and Disease, is observational, right? So you're using proxies of prenatal nutrition, things like birth weight, as I've already shown. And as I've already alluded to, there's challenges in interpreting this because genes influence birth weight, SES, all kinds of factors beyond nutrition. So semi-experiments have been really important because they're more causally informative. And one example of this is a study called the Dutch famine. And I want to know how many people have heard of the Dutch famine. I'm not going to call on you. I just want to know. Okay, it's not some, but not too many. Okay, so this is often cited as sort of the gold standard evidence in support of this model. And so let me walk you through it. This was a study that's being conducted in the Netherlands. It's not one study, it's multiple. There was a period of Nazi blockade of this region of the Netherlands during World War II. And there were records. You can see that caloric intake went way down. There was a lot of rationing of calories. And so what they've done, there have been a number of studies. And one of the strengths of this is that a number of different groups have done the same thing. So we can kind of see how replicable and how consistent it is across those studies. What they've done is they've looked at folks who were born, during that famine here to folks who were born outside that famine over here, or who were exposed to the famine in utero to those who were not exposed to the famine from the same region, that type of thing, right? So it's sort of a quasi-experiment to look at the effects of this famine exposure long-term. And when there's just a ton of coverage of this. So here's a piece from Science, mom's environment during pregnancy can affect her grandchildren. Here's a piece from Carl Zimmer a couple of years ago in the New York Times. The famine ended 7 years ago, but Dutch genes still bear scars, et cetera. There's a lot of work that's been done on this. And it's just always touted as supporting this idea that prenatal nutrition matters. But how consistent is that evidence? And honestly, I dug into this literature to try to put it together and just kind of see what is the big picture, because we all cite this, right? It's almost like a knee-jerk thing. We cite the Dutch famine as this kind of solid evidence in support of this model. But when you look at the literature, it's amazingly inconsistent and weak. And I have another version of this talk where I actually go through every slide, every study, and I go through the tables and I show you all the findings. And I'm going to spare you that and just show you the summary of that. So here's hypertension as an outcome. The Columbia study, the Utrecht study, and other studies. These are the two big ones. None of these found an effect on hypertension of being exposed in utero to famine. Cholesterol levels. So here, in the Columbia study, there's an elevated risk for some lipid measures in females, but there's equally strong protective effects in males. So very inconsistent there. In the Utrecht study, there were actually 30 different tests, and the only thing that showed up was a ratio of LDL to HDL cholesterol. Everything else did not show up. Okay, so very weak effects. Diabetes. The Columbia study didn't publish anything simple, which makes me think that they probably didn't find anything. Utrecht didn't find anything. Another study I found did not find anything. And then finally, cognitive outcomes. Columbia study, nothing. Other studies, nothing. And then in the Utrecht study, there's 1 of 30 tests that turned up yes. And now I'm going to show you, just to give you an example, kind of as a case study, look at this example of cognitive outcomes to show you how weak these findings are. This is a paper that was published in PNAS back in 2010. And they looked at all of these different combinations of things. So exposure during different periods of gestation and all these different outcomes. The one thing that punches through as significant is the percentage score on one of these tests is reduced in relation to early gestational exposure. This was not even corrected for multiple comparisons. So I don't think that this would actually hold up today. I think this got published. It may not have gotten published today. Okay, so very weak. Now let's look at this study. This is the Columbia-based study. And the authors here, they conclude individuals exposed to famine during gestation do not differ in cognitive functioning at a mean age of 58 years compared with individuals born before the famine or conceived after it, or compared with unexposed same-sex siblings. Here's another study that was published quite a while ago where they had 125,000 military recruits. And the authors of this was published in Science. They concluded the findings point either to a high order of protection afforded the fetus in utero or to great resilience of the fetus in the face of nutritional insult, or to both. And in researching all this, I found a fascinating interview with the author of this piece, Zena Stein. And the interviewer asked her, our data don't always support what we might believe is the best for public health. Has this ever happened to you? And she answered, yes, with the Dutch famine study especially. We found no evidence that 6 months of starvation and pregnancy among a reasonably well-nourished population had any effect on the intelligence of offspring. Many of our colleagues were furious with us because it would have been much more satisfactory in terms of social justice to have found that food did matter. But in fact, nothing has happened since to change that opinion. What we have learned is that certain nutrients, particularly folates, can make a difference depending on timing and outcome, but not general starvation in any direct way. Yeah. Do they have sufficient variance in BMI? Oh, you mean in the outcome? Yeah, so as adults, I think they probably do adjust for, yeah, and it wouldn't matter so much for cognitive function, I don't think, but for some of the other ones, they're going to adjust for BMI. Yeah, probably. I mean, typically you might report results with and without adjustment for BMI to see if that's part of the pathway. Yeah, but that wouldn't impact the cognitive outcome in any case. Okay, so I think we have a paradox here, right? So we know that birth weight is inversely related to adult health across the full birth weight spectrum. We also know that restricting the dietary intake of pregnant mammals, other species like mice and sheep and rats, replicates these findings. And yet here we have an example where mothers were exposed to severe famine while pregnant. And if anything, we see almost no evidence long-term for effects on those offspring, right? So I think it's quite interesting. Now, that's the example of undernutrition. Let's look at the counterexample, the flip side of this, improving nutrition. Do we see any effects of nutritional supplementation during pregnancy as an influence on offspring birth weight? And there's been lots of attempts at this through the years. Here's one study that it was a Cochrane systematic review where they took studies from the literature that met certain criteria and pooled them. And they found that in adequately nourished women, those studies, there was really no effect, sort of a 1 ounce effect that was not significant. And then when they looked at studies of undernourished women, there's a stronger effect, still not significant, close. The key thing though is if you look at sort of closer at this study, it's one particular study that's driving this increase compared to this. And that study is located in the Gambia. And this is a famous study. If you work at all in the world of nutrition or maternal child health, you'll have heard of this. This study's been going for like 70 years. It's had a seminal impact on many different aspects of our understanding of nutrition. And what they managed to do is they achieved they had a 4-year intervention where they achieved a 431 calorie per day increase across those 4 years. So it's a substantial, major improvement in nutrition for an extended period. And then they compared birth weights during the 4 years of the intervention to those in the preceding years. And they were successful in raising birth weights, okay, but specifically during the wet season, which is the hungry season, but not during the rest of the year. And so this will give you a feel for that. This is on the order of, I guess, 6 or 7 ounces of birth weight increase if they were in the wet season, but you see that there's no effect at all during the dry season. Now, what's important to note about this population is that the wet season in this sample is a period of extreme energetic stress because food is running out. Women are working in the fields, they have to, and because it's wet, there's a lot more infectious disease. So it's kind of a triple whammy. And what you see here is that during that hunger season, skin fold thicknesses, so the thickness of fat on the body, normally women are putting on weight, during pregnancy, they're actually losing that body fat, even while they're pregnant. And here you can see that weight gain is even a little bit less than 0 here. So they're actually losing weight, right? So this is severe energetic stress. And so I think that what you see from the Gambia study is that if you have a substantial improvement in nutrition, right, over multiple years, and women are, in an energetically stressful state where they're actually losing weight instead of gaining it, then you can get pretty substantial improvements in birth weight under that condition, right? But not across the rest of the year. Okay. So what about long-term outcomes? This group is, they're tapped into those same literatures about the Barker hypothesis and so on, and they recognize this as a way to try to test some of those ideas. So they looked at long-term outcomes in relation to things like cardiovascular disease risk, and they found basically no effects, right? So here's a study of cardiovascular disease, may have little effect on CVD risk in their offspring. And here's a look at cognition. We show little effect on cognitive development. I'm not aware of any study that they've found that they've published where they have found effects on long-term outcomes. Okay. What they did find is that there were substantial effects on the women themselves. Okay, so they had an increase in fat deposition, they had an improved energy level, they were more likely to sing while they were working, and so on. Like, it was notable that they were in better health. And their bodies did, in fact, allocate more energy to reproduction, but it wasn't to the current baby and the current birth weight of the current baby, it was actually to the next pregnancy. What they found is that there was a shortening of interbirth interval, right? Because when you're energetically stressed, this actually increases the likelihood of suppression of menses and so on, right? So as you become energetically better off, that's going to shorten interbirth interval, and they're not really using contraception. So there was a fairly substantial increase in the fertility rate as a result of this intervention, about a 37% increase. Okay, and if you're at all interested in this, Meredith Righteous's piece in Science from 2019 is really brilliant and worth a read. Okay, so what I think the Dutch famine and the Gambia work shows is that large changes in the mother's macronutrient intake, her protein energy intake during pregnancy, whether it's a famine, severe famine or shortfall, or a supplementation, an increase in her intake, often have minimal effects on fetal nutrition, growth, or long-term outcomes in offspring. And I think as a biologist and an evolutionary biologist, this actually should not come as much of a surprise, really, because development is extremely complex, delicate, and vulnerable. And that earliest phase of development is especially so, right? So you start off as a single cell that becomes 2, that becomes 4, that becomes 8, and then eventually all the body systems are built from those initial cells, right? And so, of course, stressors during these early phases of development could have substantial long-term and negative impacts. And so the solution to that has been to evolve buffering strategies, right, that actually buffer all that against what's going on in the outside environment. So this is a kind of simple schematic showing the evolution of some of these strategies. Fish came first, of course. They lay their eggs, in the water, lots of them, and hope that a couple survive. Very little investment at all, right? Amphibians and reptiles. You have some internal fertilization, some external fertilization. Some species have a little bit of parental care, like guarding the eggs, but very little overall. Birds, you start to get substantial control of the developmental environment, right? Because you have internal fertilization, you have eggs that are hatched, incubated by the parents to maintain a constant temperature. The hatchlings are fed and protected, and so forth. With marsupials, the early mammals, internal fertilization, live birth, and then the basic nutrient supply is the mother's body in the form of lactation, right? So there's a lot of buffering and a lot of parental care that's extended after birth. And then in us, in eutherians, placental mammals, you get the addition of the placenta to all of that, right? And so in humans, the first 9 months are a period of constant temperature, relatively stable nutrients and oxygen and other gases, protection from pathogens, not perfect, but largely. And then you have the placenta, which is invasive and gives the fetus some level of control over the mother's metabolism, which I'll talk about in a second. All right, so, but key to this buffering is nutrients and metabolism. So I'm gonna focus in on this a little bit and kind of walk you through some of this. And as a starting point, it's worth keeping in mind that every single cell in our body needs energy and substrate and proteins and so forth constantly. Like there's no cell in our body that just sort of turns off. There's a constant need for these resources to keep our bodies going. And this is maintained by a couple things, right? The fact that we have redundant sources and large onboard stores in the form of body fat and muscle and other kinds of sources, and the interconvertibility between those nutrients, right? So this allows our bodies to maintain homeostatically a relatively stable nutrient environment within our bodies. And I'm going to walk through glucose because it's crucial to fetal growth and development. So here, these are data from our study in the Philippines. We did not collect them to even look at this question. They were just sort of opportunistic. And you can see here that as fasting glucose levels in the pregnant mother goes up, the offspring, the birth weight of their offspring goes up. And we've known this forever. Like this is a very well-known clinical fact. And the reason is that as the mother's glucose concentration goes up, the gradient between the mother's circulation and the fetal circulation goes up, and that increases the flow of glucose, and that is what drives fetal growth. Okay, so let's take a look at how the glucose supply to the body is buffered and maintained. So, here you have glucose. Of course, it's influenced by your dietary intake. It spikes up after a meal, but then our bodies produce insulin and take it out of the bloodstream, right? It's used by various organs and stored. After hours, we kick into our glycogen stores, and those will last for maybe 8 hours or so. After those are used, then we start kicking into our lipid stores, our body fat. And as triglycerides and body fat are broken down, one product of that is glycerol, which enters the liver and is a substrate for producing more glucose. But the main output of that is free fatty acids. And as our free fatty acid levels go up in the bloodstream, tissues like muscle and liver and so forth will preferentially take those free fatty acids up and use them as fuel, they become insulin resistant. What this does is it spares glucose for more crucial functions like the brain, okay, which actually need glucose, and during pregnancy for the fetus, right? And then if we get really dire and we haven't eaten for a very long time, then we can even break into muscle and organs and so forth and break those down and create glucose, right? So the fetus is reliant on maternal glucose for its growth and its development. And it should be pretty clear that what the mother eats here is not the main factor that's going to drive that availability, because this is a big homeostatic system. Those dietary intakes can actually just go back into body fat stores or into glycogen stores. It's all being maintained homeostatically. Right? So placental mammals as a whole have evolved these kinds of elaborate systems to buffer early development against, the variations in what the mother's eating during pregnancy. But even compared to other mammals, humans appear to be especially effective at this buffering. And I already alluded to one of these points, right? So humans have an invasive placenta. If you look across mammals, there's all sorts of different placentas. And typically smaller species like rodents have placentas that are more invasive and that actually kind of directly access the mother's blood supply. Other large bodied species typically don't, but we do. And what this allows is for the placenta and the fetus to secrete hormones into the mother's bloodstream and manipulate her metabolism. So one of those is placental lactogen, goes into the mother's circulation. This causes the mother's tissues to become insulin resistant. This reduces glucose uptake, as I was just alluding to. And that in turn increases the delivery. So it sort of shunts that glucose to the fetus and away from the mother's body. And this is part of why pregnancy is considered a mildly diabetic state naturally, like it effectively always is, right? So that is one example of how we are different than other mammals. Another is that we have larger body size than most mammals, right, which provides greater buffering. And a big part of that is when you look at the ratio of the size of the offspring to us, that's going to be a major factor of our ability to buffer. So here, if you look at mice, the mouse litter is about half the weight of the mother, right, which is enormous. If humans had a baby that was half the weight of the mother, it would be like 50, 60 pounds, right? It's crazy. But you can see how we're going to be able to buffer a lot better than these species that have much bigger relative sizes of their babies. And a former student of mine, Zane Thayer, who's now off at Dartmouth, we compiled data from published literature to try to look at this. So the pink lines here are the Dutch famine. The Dutch famine studies sort of averaged, looking at the effects of that exposure on things like birth weight and blood pressure and glucose. And then we found animal model examples that had sort of comparable levels of dietary restriction during pregnancy. And you can see there's a clear gradient. There's a huge effect in the small mouse, kind of an intermediate effect in sheep, very small effect in humans, and so on for all of these, right? Sort of supporting this idea that we're especially good at buffering, even compared to other mammals. Okay, so I've shown you all the ways that nutrition does not matter, right? Like what the mother eats doesn't seem to actually get through to the fetus. But clearly nutrition does matter, and what aspects of it do. So let's take a look at that. First of all, micronutrients, very important, right? So these are not produced within our bodies. They're things like iron, that actually have to come from the environment. We typically have body stores, but they're limited. And if a woman is kind of chronically undernourished, those stores can be depleted. And so providing micronutrients during pregnancy often has beneficial effects, but these are very trace quantities that are required. Periconceptional nutrition, there's more and more evidence of this, like very early in gestation, nutrition right around the time of conception seems to be important. And it seems to be kind of a sensitivity epigenetically that's kind of altering the trajectory of development on some level. And we see this in these studies looking at Ramadan fasting. There's really no effect of being exposed to fasting in utero if your mother was fasting during Ramadan, unless it's right around the time of conception. You do see effects long-term in relation to that. And then really what we've known for a long time and where the most evidence is just the mother's body weight and her BMI and her nutrient stores going into pregnancy are what are important. And this, of course, is a function of her kind of chronic energy balance, her chronic nutrition over many years. And then increasingly, we're also seeing evidence for infancy and early childhood nutrition being important. And because this might be less familiar, I'm going to show you one example of that, or a couple. And we're going to use mother's leg length here as a proxy of early nutrition. I'll explain this. Right? So the hypothesis is that leg length will be a stronger predictor of offspring birth weight than will the mother's trunk length. the rationale for this is that leg growth is fast and highly nutritionally sensitive during the early infancy and childhood years. Okay, whereas trunk growth less so. It seems to be a little bit more genetically canalized, we would say, right? So this is widely used in my field as a kind of retroactive or retrospective marker of early life nutrition. And I'm going to show you a couple examples that illustrate this. So in post-World War II Japan, there was this big increase in standing height because of improvements in nutrition and in other aspects. And all of that basically was driven by changes in leg length. Right? So as nutrition improves in a population across generations, okay, you get an increase in standing height, but trunk height is staying basically the same, whereas leg length is getting longer. And this is why you can use these as sort of a retroactive marker to look at early life nutrition when you don't have the records to do so. So the main study that I work with is in Cebu, and it's a big city in the central region of the Philippines. The study that I'm going to show you here is from Antipolo City. It's a smaller study that I did in collaboration with an alum of psychology here at Northwestern about 10 years ago. His name was Green Chung. He had a connection to this birthing clinic, and he wanted to design a study, so we designed this, and he implemented it, and this is what we found. So, of course, the gestational age of the baby is a big predictor of the birth weight of that baby. The mother's BMI is a big predictor, but the biggest predictor actually is the mother's leg length, okay? Even more than her BMI and gestational age. Trunk length, less so, okay? So that's consistent with this idea that it's specifically that component of growth that's sensitive to early nutrition that seems to be predicting birth weight in the next generation. And when you look across tertiles of the mother's leg length here, adjusting for these other factors, you see there's a pretty big gradient here. It's about 300 grams or so. So to the extent that that's causal, that's a pretty strong effect of early life nutrition. And of course, it is just observational. We can't really say it's a cause, but here's something that gets closer. This is a famous study in Guatemala where they randomized 4 villages to either a high quality supplement or to a lower quality supplement. And then they sort of provided these to infants and children, looked at long-term effects, and it's the lower quality supplement villages that are serving as a control here. What they found is that intervening in diet during infancy and childhood in young girls increased the birth weight of their offspring by a substantial amount, by about 4.5 ounces. Okay? And you also see it with birth weight and head circumference. So this is closer to experimental evidence in support of that idea, right? So this allows us to return to this question, that I set out to address. Does fetal nutrition reflect weather, kind of the variability that's going on during any given pregnancy, or to climate, the sort of average conditions in that environment? And we can kind of walk through, I think, an illustrative example here. This happens to be ice core data, it does not matter. We can just imagine this as something like precipitation or ecological productivity year to year. And we can connect those dots, and we can see that it's moving around, right? But instead of connecting the dots, if we start to average across 10 time units, and then 20, and then 40 and 100, this is all very arbitrary, but the point is a simple one. As you widen the window of averaging, eventually you manage to sort of buffer out all of that short-term variability and identify that underlying trend. And it's doing a pretty good job of it right here, right? And I think that this is what fetal nutrition is doing. I've given you evidence that it's buffered against transient short-term ups and downs in the mother's intake during gestation, whether it's a famine or a supplementation. That's sort of the noise, the weather component of this. But it does seem to be tracking average conditions, right? Is experienced across her life cycle and so on. That's the signal or the climate dimension of this. And as a result of this, I think fetal nutrition growth are set to average recent nutrition, a more adaptively relevant cue for adjusting long-term biological strategy in a long-lived species like us. And I've called this intergenerational phenotypic inertia because it's an intergenerational process. It's not a genetic form of information that's being conveyed. It's actually phenotypic. And the overall effect is sort of to dampen the response to any changes in the environment based upon past experience. So there's an inertial component to it. Okay. So that's what I think might be going on in nutrition. Now, let's see if we can apply these lessons to stress physiology and the HPA axis. To what extent does fetal cortisol exposure signal or noise? And the 2 features that I've just identified that I think help kind of cue into climate is buffering against short-term variability and tracking of long-term average conditions. Like if you can find evidence for those two things, perhaps the HPA in stress physiology is doing something different or something similar. Now, but first, before we even get into this, I think it's important to point out a couple of differences in these systems, because macronutrients are resources that the body needs, right, that are being maintained within very narrow limits homeostatically. Glucocorticoids like cortisol are signals, they're not resources. And so they're inherently, they're involved in communication within the body. And they actually help coordinate those homeostatic processes, right? So they are by design dynamic, they're by design responsive. And so the stability in something like nutrient availability within the body is actually in part because systems like the HPA are not stable. They're bouncing all over the place. And this kind of illustrates that, right? So if the mother's experience is all over the place for some variable, and then we see that the fetal experience is actually not really responding to that, which is something like what we see with nutrition, what's making up the difference is stress physiology and these kinds of homeostatic systems, right? So those systems are having to be quite dynamic. And yet, despite that, okay, there is some level of buffering of the fetus. And I'm just going to walk through this very quickly. So, and this gets a little nerdy, but there's an enzyme called 11-beta hydroxysteroid dehydrogenase type 2 that's expressed in the placenta that converts cortisol into cortisone, which is inactive, right? So there's a buffer, right? So here's the mother's cortisol. Most of it you can see is being deactivated and is white here. An expression of that goes up in pregnancy as the mother's cortisol levels go up. So it seems like there's a lot of active attempt to try to buffer the fetus. The mother's reactivity to stress and things like the cortisol awakening response, when you wake up, cortisol spikes, right? Those kinds of responses are actually attenuated in pregnant women, which will also have the effect of buffering the fetus. And there's also evidence that the mother's chronic experiences might actually have influences on cortisol levels. So, things like ACEs, adverse childhood early experiences, things like discrimination, et cetera, those can actually alter cortisol levels, suggesting that, there might be some fraction of the variability in the cortisol exposure of a fetus that is tracking some aspect of the mother's chronic experiences, right? But the problem here is that the buffering of maternal stress is limited, right? So, and one reason that this happens is that the very act of pregnancy itself, what initiates it is a positive feedback that involves cortisol, and I'll just show it to you here. So you can see that all of these inputs here are positive, so that every one of them tends to accelerate things. And as a result, cortisol goes up and up and up late in gestation, and eventually that is what triggers pregnancy itself. The mother's own stress actually feeds into this and kind of can accelerate that, okay? So there's clearly a lack of buffering here because the mother's stress experience can sort of directly feed into this positive feedback and lead to early timing of parturition. And then I showed you that there is this enzymatic shield of the fetus in the form of 11-beta, right? But under period conditions of stress and other sorts of conditions, this is actually deficient. Or if the mother's surge in cortisol is very high, it's deficient. And so then the fetus gets exposed to higher levels of cortisol as a result. And then, in fact, we have a lot of evidence that acute stressors during pregnancy, okay, that are psychosocial in origin. All right, I've already shown you all this evidence that the mother's nutritional stress doesn't seem to have much of an effect, but her psychosocial stress does. And so here's some examples of that. Being exposed to an earthquake, okay, you can see that there's a big decline in birth weight, and a lot of it is driven by early gestation, just like I was talking about. Cortisol is feeding into that cycle and leading to an early gestation. Exposure to a massive ice storm in Canada, you get a reduction in birth weight. You also see a reduction in fingerprint symmetry in the baby. So fingerprints are laid down early in gestation, and the degree of asymmetry between them is actually kind of a marker of how much disruption and stress there was in utero. And you see that in relation to this, right? So that stress is getting through. 5 years later, cognitive behavioral differences in relation to this prenatal stress. And then finally, this study, fascinating. This used all births in California over a couple years. And what they're doing is what you can see is that women with ethnically distinctive Arabic sounding names had a more than twofold increase in the risk of a low birth weight baby in the year after the 911 attacks compared to the year prior, right? Showing this effect of discrimination and maternal stress on birth outcomes in the next generation. Okay, so that's my quick dive into the HPA. So let's pull this all together and wrap up. What is the potential for adaptive information transfer, in utero, and how does it differ between nutrients in the HPA? Fetal nutrition, I've argued, is largely buffered against the mother's immediate dietary experiences, but tracks her long-term nutrition, what I would call a climate signal. Whereas the HPA axis is partially buffered, but we see that there's a lot of evidence that acute stressors during pregnancy itself do impact the fetus and modify biology. Okay, so it looks like that system is more about weather. It's more about short-term variability. The implication of this for a kind of evolutionary biology is that when we think about these flows of resources, nutrient flow is more likely to provide the fetus with the sort of average conditions that it's going to be experiencing across its life, whereas stress biology is less so. It's more about sort of what's being experienced during pregnancy itself. All right, so that's one punchline. Now let's think about what all this means for policy. And there's this big push right now on the first 1,000 days. If you do anything in developmental psychology or anything in sort of global health, you will have heard about this. And if you search on it and just do a Google image search, you're going to see millions of images like this. Lots of organizations have latched onto it. This is an emphasis on the period from conception to 2 years after birth as a sort of critical period when we need to put all of our resources, or not all, but when we need to focus our resources in intervening in a way to try to improve health. And this is really derivative and kind of emerged out of this DOHAD literature that I've been laying out. And it's really, really influential. So here's an example, with the Gates Foundation. And if any of you work in have any interface with public health or global health, you will know that the Gates Foundation kind of sets the agenda for the planet with a lot of these issues. They have so much money that they devote that their criteria actually have massive implications. And here's where they lay out their strategy. We invest in nutrition to reduce preventable deaths and improve maternal and child health, with the particular focus on the 1,000-day window of opportunity from the onset of pregnancy to the child's second birthday. Okay, great goal. But I think everything I've shown you from the first half of the talk is that from the onset of pregnancy to gestation, what you provide the mother is not going to reach the fetus. This is not grounded empirically. And I think that the bigger point here is that nutritional supplements targeting pregnancy itself are unlikely to have major benefits for offspring growth or health in marginally nourished populations. I think we have a lot of evidence to suggest that. They will, of course, help the mother. There's no reason not to supplement women. I think we should supplement everybody. And more generally, I think that this DOHAD literature and the if you look at nutrition kind of holistically and how it's impacting things, it's pointing to the need for systemic improvements, more sustained improvements in nutrition, especially prior to pregnancy, across the life cycle of the mother, and so on. And I think part of what that DOHAD literature is showing is that as you improve the nutrition of a population across generations, health is going to improve. And of course, there's going to be a developmental kind of component to that. And if you look at pregnancy, you're going to see those kinds of biological changes that are unfolding. But it doesn't mean that targeting pregnancy itself is the way to improve health, right? A lot of what's going on in utero, at least nutritionally, is actually related to the mother's history, to her prior experiences prior to pregnancy. And so that is where a lot of the action should actually be. On the other hand, I think the evidence that I reviewed does align with the idea that targeting psychosocial stress alleviation during pregnancy should benefit offspring health in the mother. So if you want to intervene during pregnancy and have a big effect, try to alleviate stress more so than modify nutrition. And that's it. Okay.
How does the mortality rate affect the malnutrition? Or could the mortality rate due to malnutrition, does that affect the mortality rate? Does the nutrition does malnutrition does malnutrition factors affect the mortality rate, for example?
So are you wondering whether I'm not sure how that ties. What aspect of the talk is that tying into? Help me out.
You were mentioning in your studies, I think it was from Cebu and Antipolo, was that there was malnutrition in those areas. How does that affect the mortality rate within those populations?
Oh, it affects it a lot. Yeah, that literature that goes back for decades and decades. That's a massive effect on infant mortality. But the main factor that's actually driving undernutrition populations like that, and that leads to undernutrition and malnutrition, is infectious disease. So it's diarrheal illness and respiratory infections. Those are nutritional stressors, and they also, when kids are sick, they stop eating and they get diarrhea and so on. So those are the big factors that drive undernutrition, are the big cause of under 5 mortality, which was brought up earlier. So yeah. Yeah. And I think, so we've known that for a long time, but what's new about this field is suggesting that there are also long-term impacts of those early nutritional experiences that could affect health and maybe even the birth weight of the next generation and have these kinds of long-term effects. Yeah.
Outstanding talk. This was really, really fascinating. As you were talking, I was thinking more and more, and you addressed this towards the end about the cost on the mother of all this buffering, right? So if there is a particularly intense stress, then the mother has to kind of give much more.
Yeah.
And I imagine that there are studies about impact on mother health when they have these incidents occurring during pregnancy. And is there also, first, are there results? And second, is there a particular period of pregnancy during which that would be the worst?
Yeah, great question. So one of the implications, and I didn't really emphasize this enough, but one of the implications of these findings, or this sort of general framework, is that yes, the mother's body is buffering the fetus, but that means that under nutritionally stressful conditions, this is a stress on her and her body. And this is part of why I think you see those big effects on the mother's health in the Gambia study that I talked about. So when they supplemented the women, in that study, you didn't see any effect on the birth weights and other sorts of outcomes, but her health did improve, and her energy status and her body fat stores and her mood and all those sorts of things all improved. In general, there's a long literature going way back showing a phenomenon called maternal depletion. So if you have multiple pregnancies, okay, in a situation where women are generally sort of marginally nourished, there'll be this kind of progressive depletion of energy stores and nutrient stores that can affect her health, but also the health of her future births and so on. So, but as far as the period of gestation, when it would be the most important, I think it's going to be late because the nutritional requirements are biggest there. So early on in gestation, you've got a fetus that's one cell, 2 cells, 4 cells, et cetera. Like, it doesn't have a huge nutritional requirement. And then at some point you get a placenta and you get implantation, but even there, the nutritional requirements of that fetus at that point are probably still pretty small. But by the end, they're pretty high. So that would be my guess, is like the impacts on the mother of buffering the fetus would be biggest late in pregnancy. But I'm not sure if anybody's actually looked at that. I don't, yeah, sure. Mm-hmm. Yeah. Well, so reproductive biology has some pretty good safe holds built in. And so as nutritional status deteriorates, women will stop cycling. They become infertile, right? So that's the check on that. And the mortality has more to do with pregnancy complications more than just nutritional depletion per se.
I'm curious, we talked about the mothers a lot, but is there anything about the fathers, for example, stress in the father's life or nutrition of the father that carries over to the baby?
Yeah, that's a great question. And I think if we could come back in 10 years, we would have more data on it. There's a lot of evidence now, especially in animal models, right, like in mice, that the father's experiences can have a direct biological effect on the next generation, operating through kind of epigenetic factors that are in sperm. So if a father is stressed out, or if you overfeed them and so on, this creates these non-coding RNAs that go down to the sperm, packaged up in the sperm, passed on to the next generation, and they have effects. We do not have any evidence for that in humans yet, because no one's done the right studies. But I'm certain that we're gonna, those studies are gonna come out, and it's gonna be a big deal. Yeah. What we do see so far is that people have looked at things like when the men lose weight, or if they smoke and so on, you do see changes in those factors in sperm, but they haven't actually followed through to see what effect it has on the next generation.
Well,
It's going to be a part of the story. Yeah, I think it will be. Kuzawa.
You. Yeah, so I was also fascinated by everything, but also the Dutch famine studies, where you showed that the nutrition of the mother doesn't necessarily have an effect, right?
Yeah.
But from the extremely limited anecdotal evidence that I have from that period, I would say there was significant psychosocial stress as well. And so why doesn't that have an impact?
Yeah, no, that's a really good point. So I think there's a whole bunch of issues with the Dutch famine work. Okay. And one of them is that it's portrayed as a famine. It's all about nutrition, because the original Barker work was all about, he interpreted everything about nutrition, and it was like low birth weight babies are not getting enough nutrients and their organs are growing different as a result of that. And that was his model. And so the initial test of the Dutch famine as a sort of test of his model focused on nutrition as an interpretation. But clearly, there was a lot of psychosocial stress going on as well. And I think to the extent that you see any effects there, it could be, I think it's more likely to be driven by like psychosocial stress than nutritional stress. But there are other limitations to that study, because the control group has probably also experienced stress. I mean, even if it wasn't exactly during the famine, they're still experiencing war. Like it's not a perfect experiment. And so in some ways it's probably underestimating the effect, but any effect that is there, I would put my money down on it being driven by stress more than nutrition. Yeah.
I have a question from Zoom from Danny Abrams. He says, very interesting talk. Did nutrition affect preterm birth rates in any of these studies? Was that tracked separately or included in birth weight data?
Wait, so I didn't quite follow that. So can you say it again?
Did nutrition affect preterm birth rates in any of these studies? Was that tracked separately or included in birth weight data?
Well, typically, so some of the findings that I showed, especially those related to psychosocial stress, some of those were primarily driven by preterm, right? So like the earthquake study, you see that birth weight goes down, but it's largely because gestational age is going down. So prematurity is happening. A lot of the Doehead work, like the Barker work and so on, gestational age is adjusted. So typically, you find a stronger effect once you adjust for gestational age. And so that points to fetal growth rate as being important. And that's part of where the interpretation that nutrition is part of what's going on there. So yeah, but typically birth weight gestational age is adjusted for in those kinds of analyses.
How could we alleviate the stress or provide the nutrition requirements needed for mothers, in order to improve their health, and in case of emergencies such as natural disaster?
Yeah, I mean, I think the story that comes out of this work, there's a desire from a policy perspective to find kind of like a magic bullet or a simple little lever that you can use, like tweaking pregnancy nutrition in a narrow way and hopefully having these big effects. But I think the big story that comes out of this is that we just need to reduce inequality and actually improve the living conditions of people across generations. And this shows that health is going to improve as a result of that, right? So yeah, exactly. Child tax credit. So that's what I think this work actually points to. I think there's a lot of desire to have a simple policy solution like the first 1,000 days, like, focus everything on, on one period, and it makes it more kind of tractable and more digestible politically and more attainable. But the real message is that I think we need to reduce inequality and improve people's health across generations. And when you do that, you're going to see all these great benefits.
To remember something else that my mother told me, that when she was pregnant with me, essentially all her teeth went really bad.
But what
Her teeth.
her teeth went bad.
Yes, and so I was thinking about some other thing, which is you talk about nutrients like glucose and all of that stuff, carbohydrates, fats, proteins, but you have not talked about big things like calcium that are very important for teeth. Has that also been kind of studied, and
Yeah.
to what degree there is buffering in that?
Yeah, there is. So the mother's skeletal system is, I haven't heard about teeth. I mean, that's an interesting one. Okay. It must just be systemic, but no, there's a lot of work that's been done on pregnancy and bone mineral density. And especially in undernourished populations, you see a reduction in bone mineral density in relation to pregnancy. And that can be a risk factor for things like fractures, like hip fractures and stuff. So yeah, that is there as well. There's I focused on energy and glucose because that's where most of this field is, but you could sort of work up a whole model and framework thinking about something like calcium and how, what actually plays into that system. I think it'd be really interesting. Okay. All right.